Um, okay, so the professor has left.
And we will soon have Dr. Francisco Navarro from Spain.
We already have him. Hello, professor. Hello, doctor.
Hello.
A professor to be. Right?
Not a professor yet, only a researcher.
Work in progress.
Yeah.
All right, professor, doctor. So we are here at the conference. We're really happy that you're with us. I just want everyone to know that Dr. Francisco Juan Martinez Navarro represents the Hospital Universitario Virgen de la Arisaca. Arisaca?
Yeah.
That's the Murcian Institute of Biosanitary Research, España.
Yeah.
Okay.
And the
Gracias.
last time we met, it was in Paris, and we took a boat trip together in Paris.
Right, but you guys didn't
At the
talk.
DDX3X conference, of course.
Yeah.
And my husband was with us as well.
Yeah, but you guys didn't talk to each other yet.
No, I was too ashamed to ask questions.
Ursula
I had
was
so
shy.
many questions, to be honest,
Ursula,
to you, Francisco.
can you come closer so
Yes.
that Francisco Juan will see you?
By that time, I had so many questions to you about the inflammation, about, interesting Polish case we have, but I'm sure we will be able to catch up on that.
Yeah.
But I'm glad you are here with us and you agreed to take the invitation.
Yeah, it's a pleasure to.
I was more brave over an email, to contact you.
Good that you were. Francisco, if you can keep your presentation to about 15 minutes, that would be wonderful.
I will adjust to that. No problem.
Okay, thank you so much. And you can start now.
Okay. So, as they present me, thank you. First of all, thank you for the opportunity to present our work here. We are working in Spain, in Murcia, in the hospital, and we have a group in which one of the person that is involved in the project is hired with the money funding from the DX3 Association from Spain, that is Esther Gomariz, that is the person that is in charge of the screening and the experiment now. And I'm going to show you how we are doing these therapies for drug reperfusion work to try to find treatments for DDX3X syndrome. First of all, my presentation is a little bit different from the previous one because we are not working with the human data directly. We are modeling the disease in our animal model, that is the zebrafish. That is this small fish that is around 3 centimeters when they are adults. But what we use usually for doing our experiments, they are the larvae that they are obtaining from the fish every week. One of the main reasons why we are using fish instead, therafish instead mice or other models, is that we can obtain a high number of embryos that can provide us more information about the treatment that we are trying. So we can obtain around 200 embryos per couple every week. And one of the best things about therapies is that the development is really quick. So in 24 hours, we have the larvae almost completely formed, and we can start to do treatments at that state, at 1 day post-fertilization. And as soon as 3 days post-fertilization, we can test and check the effects of these drugs. And also, they are transparent and they are really easy to modify genetically. And this is a huge advantage due to the the high throughput screening necessity for a high number of embryos, and to modify the fish to obtain the model that we are looking for. So you could be thinking, zebrafish is not so similar to a human. How is possible that this is resembling the disease? So zebrafish are inside the vertebrate group that is super close to the human or to the mammals also, and they are studying mostly the complete organs. They have the same structure in the brain, eyes, ear, heart, liver, intestine. So they also share around the 70% of the genome with the human and the 85% of the genes that are related to disease. So this is providing us a very good tool to reach our objective to obtain some treatment or drugs candidates to be used. As previously mentioned, the DDX3X gene has different domains, and there are several mutations in these domains. So there are different types of mutations. The missense are the frame seed, and in our case, in zebrafish, the homology with the protein is around 70%. That is pretty similar, and the structure is almost the same. And one of the things that we are doing now is using our model blocking the DDX3X gene. So we are in work to obtain different mutations to check the different ways of the missense frame seed. But at the moment, we are using a model that is blocking the DDX3X genes in zebrafish. So to do this, we are following this kind of workflow. So first, we know which are the mutations in the patient. So we are trying to resemble that in the zebrafish. And after obtaining this model, we are trying to test, we characterize it, and we are trying to test different drugs and check if these markers that we characterize it are changing and are coming back to the normality. And in this case, we try them in different ways, and at the end, we can propose them for drug repurposing that should be validated in other models, but this is accelerating a lot the process for a new drug application in any disease, because these drugs that we are testing, they are approved by FDA, or they are natural compounds that can be used without spending the 20 years that usually takes the clinical trials. So first, to obtain the model, we inject in the first cell stage embryos. So we inject the CRISPR-Cas9 sgRNA that in this way we modify the DNA, inducing the modification of the DDX3X gene and blocking their expression. And when we do this, we characterize the model, and at 3 days post-fertilization, at 72 hours, we started measuring the larvae, and we observed that they were smaller than the control one. Also, we checked the movement of this larvae with an assay, and we observed that they also move less than the controls. And all of these could be used as a marker, but these are really time-consuming, so we were looking for another very quick and fast marker to analyze. And we also, in our group, our expertise usually, or my expertise, was the inflammation. So I was using this DDX3X modification in some fish that has cell-innate immunity cells, marked in green, to observe the effect of this mutation in the immunity. And we observed that the neutrophil counts increases. That means that there are more immune cells or innate immune cells in the DDX3X mutant. But when we were developing an assay to see the resolution of the inflammation, we observed that the resolution was delayed in these mutant fish. That is indicating that there is more immune cells, but they are not competent enough to to do their work to resolve the inflammation. Lastly, we found another three markers that are the Wnt activation, that is one of the main pathways in this fish, and also the NFKB. And we observed a really constant change that indicated a decrease in the inflammation, these two inflammation marker, and also another gene that is related to inflammation and signaling of the inflammatory response. So with all this data, we selected which were the best markers to be following the following screening of the drug, and we selected the NFKB because we could be observing this easily, and also we could automatically measure it after doing the imaging. In this way, we could be going faster. So after having the model characterized and selected the marker that we are going to be using, we started the drug screening. And at the moment, we have tested some compounds already that are from a natural compound library. And usually, this is our expectation, is that around 2% of the drugs that we are testing, usually in the previous drug screening that we have developed, that 2%, around 2%, are going through the next stage because we observe that they have the effect that we expect. And after a second round, that means that we are checking other markers. Usually, this is reduced by half. And usually, at the end of the drug testing in other models and everything, there is only 1 or 2 drugs that are proposed for the report producing. So in our case, this is the workflow that we are following. We block at the beginning the expression of DDX3X, and 1 day after, we use the different treatments, and we image them 2 days after to check the effect of these treatments. And this is a more elaborated one. We also not only image them, we collect them to see different gene markers also. And at the moment, these are the results that we are obtaining. In green is the normal level of the NFKB activation in control, and in red is the levels of the DDX3X mutant that we have. And here you can observe all the compounds level of NFKB in the DDX3X mutant, and how they can, some of them are inducing more even activation, NFKB activation, others are inducing a little bit, and others are reducing. So from this huge amount of compound, we are selecting the one that have similar activation on NFKB of the controls, and we are going to the next stage with them. In this case, I can show you, for example, how they look, these larvae. So the controls usually are pretty bright, and the DDX3X mutant, they have very low intensity. So we are measuring these intensities, and we are, for example, in this case, there are 2 compounds that they are two hits that we are going to use in the following rounds that can recover the, the activity of the NFKB. This is not means that it's going to recover the control phenotype totally. It's only one pathway. This is the reason why we are doing several rounds. So after this round, we are going to test another markers to see if it's changing different pathways. And now we are in this couple of years or year and a half, we have not only started doing this screening, but we have started developing a new model that is stable, that we don't need to be injecting every time to block the DDX3X expression. So we have achieved DDX3XB because in geographies there are two copies of this DDX3X gene. So we have observed that when we block only the B form of DDX3X, and we have obtained a similar result or even more consistent than when we are doing transcellular. So now we are doing some experiment with these fees that, for example, we have sent some samples to obtain the gene expression profile. In this way, we could be able to obtain more markers. We will be using this new model probably in the future to be going faster and to obtain consistent results and making more accuracy drugs test. That's all from my side. So this is my group. This is Esther, that is the lab member that is doing the experiment now. And this is my PI, Maria Luisa Cayola, and we are happy to receive any question from you or any comments. Now or later, you can contact us directly by mail.
Thank you, Professor.
Thank you.
Let me see if there are any questions here in the room. Yes, we have one. You will pass the microphone. Before that happens, I just wanted to show you, Doctor, that we're very proud that the Spanish representation is with us. So here's the flag, we got it. Okay, and here's a question from Demis. Okay, maybe you just need to, yeah. Okay, so why don't you take this one?
Hello, Doctor. Can you hear me?
Yeah.
Yeah. Just a quick question. If I, because I probably didn't understand exactly the purpose, not the purpose, but if, let's say, you develop this drug, is this drug would be used before, like during when the yes, during pregnancy, or could it be used like after when the baby is born and during its life, for example?
Sorry, I cannot hear you very well, but I think that I understood the question. You are asking if we are trying to do this reproducing for very latest, very early stages or later in the life?
Yes. So is it going to treat the already the person that already has the condition, or before it's born?
Yeah. This is one of the things that are very important to take in account, because there are some structural changes that probably are not easy to resolve, but there are other symptoms that can be dismissed with the treatment in later stages. So the optimal case is that we are, in our model, we are doing it in very early stage. And this is going to be like informative, but not fully, not the full information, because we are going to obtain some results that probably in the future, if the treatment that we test are reaching the patients, they are going to obtain some benefits, but probably it depends on the states that they are administered to the
Demis,
passing.
Demis, did you get the answer you were looking for? Yes, we got the answer. Francisco Juan, thank you so much. And we have one comment from Elena from Spain. Congratulations, Francisco Juan and partners. Asociación DDX3X España is proud to collaborate with you.
Thank you so much for the DS3X Spanish Association.
Of
They are
course.
our engine.
And now Professor Roxana Malak from Poznań.
Hello, thank you for your lecture. I would like to ask, did you found any special things after putting the drug to the fish, referring to notochord, is something like spinal cord of the fish, you see? Because, inhibition, the inflammatory, when you would like to inhibit the inflammatory process in fish, did you see any changes in notochord or something like spinal cord of the fish? Sorry? Or, yeah. Referring to neuroinflammation, you see, which can be mentioned in children with DDX3X.
Yeah, what we have observed in our model is that some inflammatory markers that we have checked, they are low in the mutants. So in the previous conference that I attend, I remember that some of the families were talking about their child could have more infection and all these things that they correlate with our results that we are showing that there are more innate immune cells, but they are not good enough to resolve inflammation because probably the DDX3X is involved in some pathways. Probably no, it's published that it's involved in some pathways for inflammation. One of the things that we are not doing, because we are doing it blindly, is we are not selecting drugs specifically for something. We test before some drugs that could be modulating this inflammation, but at the moment we are doing the drug testing screening with the whole library from the natural compounds and FDA approved library.
Okay, thank you.
And we have one more question.
We will soon have a meeting with Maria Cayuela, the supervisor of Francisco.
Okay.
So we will discuss it in more details.
Oh, that's good.
Hi, Juan. Thank you. Just a question. How many drugs have you screened? And in the library, you mentioned how many are left to be screened. Like, what's the timeline to screen all the potential drug candidates?
Yeah, the libraries that we are using, each of them have around 800 compounds. We have tested around 200 already, and all of them are suitable for drug repurposing. I mean, they are already approved by FDA, or they are natural compounds that can be used after these different stages of analyze. So I don't know if I explained it like in the best way, but after our screening, we are doing a second round in the therapies also. But after just confirming the effect of some of the drugs, we are going to be doing it in different models just to confirm that. And in that way, we could be like showing more powerful results to be proposing some drugs for repurpose.
Thank you. Thank you, Francisco Juan. We're happy with the answer. And then also we will hear from your supervisor, which is good as well. Thank you and have a great rest of the day.
Thank you so much for everything. Nice
Thank
day.
you so much.
Thank you. Keep up the good work. All right. And now we'll take a 30-minute break,
And before
which is
that-
well deserved. And before that happens, Ursula has some really good news.
So we have a surprise for all of the parents because we meant to have a raffle at the end of the conference, but Młyńska 12, which is a great and unusual place
Like
in
a
Poznań,
conference and dining location. Pretty
That's a
fine.
very original place.
I organized my wedding reception there.
Well, I've organized my wedding reception there as well.
And we are not married, so there were a
We
lot
are
of
not.
receptions.
We are not, but it was two
Maybe it
wedding
was,
receptions.
yeah, maybe we are. I don't know. I'll need to
So-
check. Let's
So-
talk about it in the coffee break.
Yes,
Yeah.
let's do it. But we have a gift for you, for all the parents and to all the children with DDX 3X. We are inviting you to Młyńska 12
To a
at
dinner
to a
tonight.
dinner tonight, 7 p.m.
Everybody.
Everybody.
Yeah. So see you in about 30 minutes. If we make it 25 minutes, that'd be even better because we are there good on time. And the first presentation after break will be in Polish. So you might need your earpieces. Yeah. Or you just learn Polish on the go.